Hello friends, my dear students, myself Wandhare Sheetal from XII.SCI Rao Bahadur Narayan Rao Borake College Sirampur. This video is prepared under the guidance and support of Honorable Principal Dr. Sahib. This video is only for the educational purpose. As per here the new syllabus of the State Board, the topic that is the human health and diseases, shows the content that is the immunity, the definition, types of immunity, how the working of the immune system takes place in our body, which types of different cells helps in this immunity system, the structure of antigen antibody, their complex formation, and in the second half part of this chapter here, the different types of the diseases that we are going to show. Now here let's start with the concept of the immunity. Here, as we know that here our body itself is having the capacity to fight against the diseases, and this capacity, it may be here the inborn capacity, or that capacity is get produced by injecting some medicines, syringes, or preferred by the pathologists, doctors, etc. Now here, what is this immunity? This immunity, it is nothing but the resistance power of a body to fight against the different types of the diseases, that is the immunity. And here, to increase or to maintain the immunity, our health, it should be in the normal state. And what is this health? This health is nothing but here the completely merely an absence of a disease, or it is the complete social, mental, physical wellness of the body. It is considered as the normal health. And if we are suffering from any type of the diseases, at that time body first trial itself automatically get repair. Why this is takes place? Because the immunity system, it is playing the very important role in our body, and here this immunity system is to fight the resistance. Now this chart is showing two types of the immunity is present in our body. Here, that is the innate immunity or the acquired immunity. This innate immunity means the immunity which is present from the birth. From the birth up to the death, these different types of the immune systems, they are working against the different types of the pathogen that comes under the heading of this, the innate immunity. But here, as we know that here, each scientific word has evolved from the Greek as well as this Latin language, and how that immunity system or how that concept came in existence, the firstly here that shows that when the Edward Jenner, they are created the vaccines against the different types of the viruses. The smallpox virus first he have taken into trial, and when he tried on the human body, he get the result that the person itself get the safe and cure. Then he notified that these vaccines, they are get helpful to kill that such type of the virus, and until and up to some diseases they are itself curable. Therefore, that immunity, they will be get identified that two types of the immunity is get come in our body. And this immunology is nothing but here, it is the branch of science which deals with the study of the immune system in response to the foreign substances and their role in resisting the infection by the pathogen. That is the immunology. This immunology, it shows the study of the immune system and the different types of the antigens, they are present on the surface of the RBC, as we know that. And in response to the antibodies, here in response to necessity, these antibodies, they are get produced. Here that antigen is nothing but what? Any foreign substances invading the body and capable of stimulating an immune response is known as a antigen. These antibodies, they are nothing but here the protective chemicals, and these chemicals, they are get produced in immune system in response to antigen. When we required that antibody production, then only these antibodies, they are get produced. For example, here the XYZ type of the virus bacteria is get entered into body, and whatever the antigens, they are located on the surface of RBC, here they will be get notified that to produce the antibody, because different types of the antigen antibody molecule, they are produced in our body during the each second, and here to respected fight against the viruses, bacteria, these antibodies they are get produced. When antigen, they will be give the responses, they will be give the signaling. Here this innate as well as the acquired immunity come towards that the immunity which is present from birth, that is known as the innate immunity, and the acquired immunity, this immunity is not present from the birth, it is get developed artificially by injecting the different types of the vaccines, medicines, syringes, etc. Now this innate immunity, it follows the different types of the barriers, and these barriers, they are the present by birth, they are not invading outside. For example, here this skin is present in our body by birth. Different types of the blood cells, they are present in our body by birth. The cellular factors, why this fever is produced, how these acute phase proteins they are get produced, that things they will be produced in our body by birth, and therefore this comes under the inborn factors. Now we call them as nothing but the barriers, because these barriers, they have to prevent our body from any type of the infection. Let's take here the first one, that is the epithelial surface, that is nothing but the skin. Epi means the above, and we know that here the skin plays an important role in fighting the various types of the diseases. How? The very simply, the transpiration means homeostasis, means maintain the body temperature. Automatically this all the functioning takes place with the help of the skin. That homeostasis, it will be takes place with the help of the skin, and these healthy skin, they possess the bactericidal activity due to presence of the high concentration of the salt, as well as by secreting the sweat, because the sebaceous gland and sweat gland, they are located on the skin surface, and therefore by secreting here these sebaceous secretion, the long chain of the fatty acid, they have the bactericidal and fungicidal property. Due to that, the mucus of the respiratory tract also here, it prevents the entry of the microorganism. Whatever the dust particles, they are get entered inside the nose, here the hairs are present on inner lining, and indirectly here these hairs, they get trap the dust particle, as well as here they trap the foreign particle and allowed it to prevent entry in the respiratory tract, also in the nasal chamber. In this way, here the epithelial surface, it also plays the important role. Suppose the cough reflex, it is also the important mechanism of the respiratory tract, because the mouth, it is the constantly bath in the saliva, and here which has the inhibitory effect on the microorganism, the acidic secretion in the stomach, it is also the example of that surface, because the acidity maintained in the stomach region to kill the microorganism, which microorganism may enter in the digestive tract through the food material as well as through the water, and these substances, they are automatically get killed if along with that substances such type of the viruses, bacteria, pathogens enter into the body. These viruses, they will be killed because of the secretion of the one normal HCl present in the stomach region. Here the secretion in the vaginal tract also prevents the entry of the bacteria. In this way, that epithelial surface which present by birth prevent the entry of the foreign particle. The second is the antimicrobial substances which is present in blood and the tissues. These antimicrobial substances means thereby about the 30 different types of the serum proteins present in the circulating in blood, and these are in an inactive form present. The presence of microbial pathogen, when they enter, they activate these substances and the complement cascade to eliminate the pathogen. The interferon are class of the cytokines released by virally cell infected with viruses, certain WBCs, to stimulate the other cell and to prevent themselves from the viral infection. The third is the cellular factors. Here the cellular factors that natural defense against the invasion or blood and tissue by microorganism along with the other foreign particle is mediated to a large extent by the phagocytic cell which ingest and destroy them. That phenomenon takes place with the help of the lysosome, because these lysosomes they are having the phagocytic nature, and due to this phagocytic nature the engulfing of the microorganism takes place, and these cellular factors plays the role to kill the virus, to kill the bacteria or the pathogen. After that, here the fever, that fever when comes inside the body, they are showing that here when any such a type of the infection is there, at that time automatically body temperature goes on increasing. The rise in body temperature, they will be shows that XYZ type of the infection is takes place in the body, and that such infection, it indicates that, or the increasing body temperature indicates that your immunity system is properly working, because here all the WBC cells, they are helps to play an important role in the immune system, and this body temperature increases indirectly here, that helps to accelerate the physiological processes that destroy the pathogen, and this fever stimulates the production of the interferon which helps in recovery from the viral bacterial infection. Means here that fever, it is showing that the proper working of the immunity system, and which are types of, or which are different types of the immune cells they are present in body, they are working in such a good manner. The next is the acute phase proteins. This acute phase protein is nothing but here the infection and injury which leads to a sudden increase in concentration of a certain plasma protein, collectively called as the acute phase protein, and these include the C-reactive protein, mannose binding protein, alpha I acid glycoprotein, serum amyloid P3. These different types of the proteins they are working here, and they helps to increase the resistance power, because automatically here they prevent the infection from the any type of the pathogen. Now here come towards the acquired immunity. Here this acquired immunity, it shows that the two types, that is the passive acquired immunity as well as the active acquired immunity. This active acquired immunity is divided into natural acquired active immunity and artificial acquired active immunity. Then this passive immunity is divided into natural acquired passive immunity as well as natural acquired here the passive immunity. Okay, here both they are having the natural as well as the artificial, and which type of the cells, how they are working, now let's see that. But firstly here come towards this acquired immunity. This acquired immunity, it is the resistance of an individual that acquired during the life, and therefore it is get acquired by taking different types of the vaccines, and these vaccines they are ready to create that type of the antibodies throughout the lifespan up to the death of that individual. For that purpose here different types of the vaccines they are get given from by birth. Now these acquired immunity, they are having the specific features. Now which are the specific features of this acquired immunity is present? Here they are showing the first, the specificity. It is also the important feature of this acquired immunity. That specificity means here it can produce the specific antibody or a T lymphocyte against a specific type of the antigen or against the specific type of the pathogen. Therefore, that is the specificity. The second is the diversity. it can recognize a vast variety of the diverse pathogen, means the different types of the viruses, bacteria present in nature, and these cells, they are having the capacity to identify different types of these pathogens. Therefore, here this diversity, it is also a unique feature of this acquired immunity. Then third is nothing but the discrimination. This discrimination between the self and the non-self, means here that cell is having the capacity to identify our body cell and to identify the foreign particles or the foreign cell. This WBC is also having the capacity to identify our cell, as well as if any foreign particle is entered, immediately they are start to working, and the phagocytic role, it will be takes place because of here the identification or the discrimination between the self as well as the non-self. And here the fourth character of this is the memory, means here once the immune system, it get kill any type of the pathogen, and again twice that same pathogen is get entered inside the body, immediately these immune system, they will be get identified. Their first encounter, they will be get memorized here because of they are having the capacity to identify, because of they are having the memory. Suppose here the individual is suffering from the typhoid, and again twice that same person is suffering from this typhoid. Very first trial, the body is getting identified that Salmonella typhi, and he has to cover itself trial. If not get cured automatically, then we are get preferring to get the advice, we are get preferring to the extra medicines to get killer. Okay, this is about the specific features of the acquired immunity. Now this active here, acquired immunity shows natural acquired active immunity. What is happening in this natural acquired active immunity? Due to infection, here that immunity is get acquired and it is developed after the entry of pathogen inside the body. It is long lasting immunity, and this immunity, it is when the person is recovered from the attack of the measles, the developed natural acquired immunity, and this will be happen for the lifetime. The second is the artificial acquired active immunity. It is the resistance induced by the vaccine. This vaccine is introduced into the body for getting to stimulate the antibody in response to the immune system. For example, here the BCG vaccines, polio vaccines, they will be given to the children at the birth, and throughout the lifespan here these child, they will be get protected, because if that type of the virus get entered, that polio is the virus, BCG vaccine, they will be against to get kill that such type of the pathogen here. If that virus is get entered, already that child has given that vaccine, and therefore automatically the entry of the virus, that will be create such a type of the antibodies, and that will be get killed here because of this immunity is get developed in this children. Now this passive acquired immunity, it is again divided into natural and artificial. Now this passive immunity is acquired when body it is get the ready-made antibodies, and they are received by the body cell, that shows this passive immunity. Now in first type, this after the birth, when the maternal antibodies, they are get transferred from mother to the child, that may be get transferred through the placenta, or that may be get transferred through the mother's milk, and that mother's milk, it contains the colostrum, because that shows the different types of the antibodies, and that will be gives here power to that fetus to fight the diseases. These antibodies, they are received from the baby and from that mother, but these antibodies, they are not remain for a long time. They are remain in the body of that children for the very short duration. Therefore, this shows that is the passive immunity which is lives for here the very short duration. The next is here the artificial. This artificial acquired passive immunity, it is developed by injecting the previously prepared antibodies using the serum from the humans as well as the animals. And these antibodies, for example here, the different types of the antibody serum, it will be get produced from the animals. Now here the horse, dog, in case of the rabies here, the dog serum is get used. Here the horse serum is also get used to get produce different types of the antibodies, but this is not for the long life. These are get produced, these antibodies, for the very short duration to kill or to cure that such type of the infection is there. Okay, now here come towards the cells of the immune system. In these cells of the immune system, the two types of the cells, that is the lymphocyte as well as the antigen presenting cells, these cells they are involved in the working of the immune system. Now come towards here the lymphocyte. These lymphocytes are of two types, B as well as the T lymphocyte. These lymphocytes, they are the major cell, plays an important role in the immune system. When we have shown the classification of the WBC, at that time here that lymphocyte, monocyte, they are plays the important role in the phagocytic function. Here these cells, they are get produced in the stem cell and the hematoblast prevent in the liver cell of the fetus as well as in the bone marrow of the adult. The some of them undergoes differentiation in gut associated with basal lymphoid tissue, that is the tonsil, Peyer's patches, and they are called as the bursal or the B lymphocyte, and the other cells they are get differentiated into the thymus gland, they are called as the T lymphocyte. How this T lymphocyte and B lymphocyte is get working? This mechanism of T lymphocyte, it follows here the 4 stages. That is, in the first stage here the helper T cell, but when the any type of the pathogen is come in contact with the T lymphocyte and they forms the clone of the T cell which are similar but they perform a different function, and these clone has the 4 types of these T lymphocyte. The first one is the helper T cell. These sensitized helper T cell, they are produces the lymphokinesis for performing the different types of the functions, like that is the stimulation of the B lymphocyte as well as the macrophages in the killer T cell or the cytotoxic T cell. They get directly attacked and destroy the microorganism. They are the infected body cell, just like the cancer cell, the killer T cell, they are bind and infect to secrete the porphyrins. These porphyrin form a hole in infected cell and they also release the substances to kill that such type of the cell. Hence, they are having the name the cytotoxic T cell, because that type of the pathogen virus, it has to get killed by secretion of the such type of the substances. Now the suppressor T cell, their meaning is showing that it will be get suppressed, and these cells suppress the entire immune system against the attack on the own body cells. They have to get protect the immune system. The memory T cell, it shows that the previously sensitized cell which retain the sensitization memory for the long time in the future, because any type of the pathogen is previously entered and it will be get encountered. Twicely that pathogen, if it is entered inside the body, automatically that will be get identified because of having the memory. Now the mechanism of the B lymphocyte, these B lymphocyte are sensitized directly by both antigen as well as the helper T cell, and activated B lymphocyte, they multiplies because they have to produce the clone of the plasma cell and the memory B cell. Here the first is the agglutination. In this agglutination here, the particulate matter, that is the bacteria, viruses, they are mobilized and the mass it is get formed by the process of the phagocytic, it will be get engulfed, that forms the agglutination, or this agglutination also forms the antigen antibody reaction also. The second is the opsonization. In this here, the coating of the bacteria to facilitate their subsequent phagocytosis by the macrophages. This shows the opsonization. Means here due to again the phagocytic movement, these macrophages they will be get produced. Neutralization, it is the next step of the B lymphocyte. In this neutralization, the toxins they are get released by the bacteria. For example, here the tetanus toxin, and each antibody, as we know that here it is the produced against in response to the antigen, and this antibody it is having the specific, they are because fixed in the lock and key manner, they are having the specific antigen binding site. Now the second cell of the immune system is the antigen presenting cell. This antigen presenting cell again they engulf the invading pathogen by the process of the antigen, and they process this antigen represented on the surface of the RBC. These cells they are able to deliver a stimulatory signal that is necessary for the activation of the helper T cell. This antigen presenting cell they are located on the surface of the RBC, and here they will be get signal to the T cell. At that time the mechanism of the T cell it will be get started here. Okay. In this way here we have seen about the definition of immunity, health, different types of the immune system, then how this immune system is get working inside the body by producing different types of the antibodies in response to the antigen. Okay, and the mechanism of the T lymphocyte as well as the B lymphocyte. Thank you.